Okay, so welcome to our presentation. My name is Liang Huang from Applied Research USA and Oregon State University. Today, I'm going to tell you about our recent work on mRNA vaccine design using computational linguistics. So this is, some work that we developed from computational linguistics insights and inspirations and kind of adapt those algorithms and concepts to computational biology to design mRNA vaccines sequences that are optimized for energy and the codon optimality. Okay, so as you all know, this pandemic has completely changed the world, and to stop it, mRNA vaccine has emerged as the fastest and arguably also safest vaccine route among all vaccine types. And we have already seen some preliminary results, very promising from many different companies, including Moderna and others. But there's still a long way to go before you can use it for the general public. And many challenges remain. And probably the most important one of them is chemical instability. Because RNA vaccines are single-stranded mostly because it's RNA instead of DNA. And because it's single-stranded, it's not very stable. It's easily degraded by water molecules or enzymes, and you could be cut into pieces. So our solution is to use computational linguistics algorithms to design more stable mRNA vaccines, which can hopefully produce more proteins or the antigens, which can trigger more immune response. Okay, so as I said, there are three major types of vaccines: protein, DNA, and mRNA vaccines. And among them, protein is definitely the most dominant and the most commonly used one, for example, the flu shots. But it's very slow for mass production and has a small risk of infection. So this is probably not the ideal solution for this pandemic. So we need much faster mass production vaccines like DNA or mRNA vaccines. And we also prefer the mRNA vaccine because it has no risk of integration into your genome because translation happens in the cytoplasm rather than the nucleus. So it's the safest. But the drawback is that because it's single-stranded, mostly, it's very unstable chemically. And before it can be translated into protein, it can already be cut into many different pieces, so it's no longer effective. So our question is, how do you design more stable mRNA sequences so that we can solve the chemical stability problem, and that there is a related problem of immunogenicity, that is how many proteins or the antigens you can produce or translate into. So we want the more the better. So we want both stable and high protein yield. So yeah, that's the goal of our talk. And let's look at this problem a little bit more in detail. So there is a redundancy in the genetic code that each amino acid, as we all know, can be translated by multiple DNA codons or RNA codons, the triplet codons. For example, alanine can be translated by 4, arginine by 6, and so on and so forth. So if we look at each protein sequence, like for example, the spike protein of the coronavirus, which is the common vaccine target, we got like 1 choice, 2 choice, 4 choice, blah, blah, blah. We multiply them all together. We got 2.4 times 10 to the power of 600 something, which is a humongous number, and you can never enumerate them one by one. So there are so many different sequences, and each one of them translates to the same protein. So they're all synonymous mRNA sequences. So our job is to find the best one out of this sequence, out of this huge space. But how do you, what's the criteria you want to choose from? Well, Moderna's paper in last year's PNAS journal suggested that the more stable an mRNA is, not only is it more stable, but it also leads to more protein yield, which is reasonable which is intuitive because the more stable you stay longer in cells, there's more chance you got to be translated into proteins. If it's unstable, then it can easily be cut into pieces. So before you get translated by the ribosome, you already become subsequences, no longer effective. So now the question is, how do you find the most stable sequence in this gigantic space? And our answer is to use dynamic programming plus formal language theory. And the key idea is to represent mRNA as a DFA, or that determinate finite state automaton. And for example, the valine, each amino acid is represented by a small finite state automaton. Valine has 4 choices, you represent that this little graph. Serine has 6 choices, you represent like this. And then you concatenate all of these little DFAs together. You got a very big, very long protein DFA. And now you suddenly realize that each path in this very long protein DFA corresponds to an mRNA sequence that can translate to the same protein. So this huge 10 to the whatever 600 something space is now packed in this very small DFA form, which represents exponentially many different choices, right? And then we use an old technology that used context free grammars to represent RNA folding free energy model. So this is very, very old. But now the crucial insight comes that we realize that mRNA design problem can be reduced to an old problem that is CFG intersecting a DFA, context free grammar intersecting a DFA, which is a very old classical concept in computation theory and computational linguistics that is taught in all the every undergraduate CS curriculum. But this insight is the new contribution here that nobody has to look at this problem from that angle. But once you realize that mRNA design is just CFG intersection with DFA, then the problem is very simple. You can solve it in cubic time, where n is the RNA sequence length. That's the dynamic programming algorithm. So we implement that algorithm, which is the exact search algorithm, runs in cubic time. It's pretty fast, but we can even make it faster by using linear time approximation using LinearFold, our previous work. And for spike protein, the n is roughly speaking 4,000, less than 4,000 nucleotides. The wild type is pretty bad in energy. And with exact search in an hour, we can improve the energy by a lot. You got a very, very low energy, like negative, 2,000. And with approximate search, you can do much faster, 11 minutes. And the energy difference is very, very small. You can get almost as good as exact search. Still a lot better than the wild type. And you can compare the wild type and the linear design structures. The wild type has a lot of unpaired regions, like these little loops, which are easily being cut by enzymes or water molecules. So it's very unstable. Whereas our linear design sequence structures are almost like DNAs, almost like all double-stranded, right? So there's no pieces that can be easily exposed to degradation. But that's not the whole story. The other half of the story is that the Moderna paper also shows that codon optimality is equally important, if not more important. So you need to be good in both codon optimality. You need to choose very frequent codons and also be good in energy. So we successfully integrated codon optimality, or CAI, codon adaptation index, into the joint optimization. So we can do it together in the dynamic programming. And then we can do a two-dimensional optimization. On the x-axis, we want the left the better. I want lower energy. On the y-axis, we want higher CAI. So this is the best we can do. This is the feasibility curve, the limit. Beyond which we can no longer, there's no sequence that is possible in this region is infeasible, but everything here is feasible. Whereas the wild type is very bad in both energy and CAI because the virus has just recently jumped from another organism to humans, so it has not got enough time to adapt to the human codons. So with time, like say in 50 years, we will see the coronavirus probably end up in, roughly speaking here, it's got better energy, but better CAI. And if you do the CAI greedy design, that is for each amino acid, you choose the best or most frequent codon, then you got CAI equal 1, and that's this point here. But it's much better in energy, but still not very good in energy. We can do much better than that. Moderna's paper did not explore anything here in this region. That is a vast uncharted area that nobody has touched before. So whether this region will produce better vaccines, that's a question that we need to be seen in real wet lab experiments. So with that, I would like to thank you, and please try our web server, and you can check out our preprint. And the web server, you can input any protein sequence, and we will give you the best mRNA sequence. Thank you so much for your attention, and hopefully we will have a vaccine very soon.